All right. So while Valerio gets set up, nice to meet everybody. So I'll give a full disclaimer. We're not going to show any data on CHAMP1 because we actually just got funded. I actually got to join the community about 2, 3 months ago. We met Jeff about a year ago. He cold emailed me. He kind of said, come to a call, and eventually we kind of got talking, and that kind of led to the kind of the stuff we're going to talk about today. And I'm gonna, for the first 2 minutes, I'm just going to introduce what I do in my group, and then essentially Valerio is going to do all the hard work. So any questions, go. You can go to town on them, please. Uh, so essentially, you're kind of probably wondering, how did an Irish guy and an Italian guy from Montreal actually end up here? And I'll explain why. I oversee a group in Montreal, it's the Neuro's Early Drug Discovery Unit, and this just gives you an overview of the kind of process. We're based in Montreal. We're at the Neuro. It's a neurological institute, research and clinical hospital. It's been going over 90 years, founded by Walter Penfield. This is winter time. You're welcome to come. It's nicer in the summertime. And about 10 years ago, we actually set up our own platform, focused on induced pluripotent stem cells, or iPSCs, because I was working on Parkinson's, I was working on brain disorder, but we were working with HeLa cells, HEX cells, cancer cells, mice models, and we were kind of wanted to know, could we really work with human brain cells in addition? That led us to form what we now call the Neuro's Early Drug Discovery Unit, or EDDU for short. We're kind of a full circle group. We're actually downstream. We have two groups. It's myself, our group, Open Science Group, and we also have CBIG, which is now the Neuro's Open Biobank, which kind of lets us recruit patients, consent them all in an open access manner. We get the blood from them, skin, urine. We program them into iPSCs. We can then CRISPR edit the lines and then make the cell types we would need to actually then study them, maybe make a discovery, figure out if a gene X has a mutation or it's absent. Is there a difference between it and the control? And we've built all the tools and team to actually then do this. So for the talk really today, we're really going to focus on the 2D and the 3D part, and that's really where Valerio is going to come in. So how do we work with others? We've been going 10 years. We're actually not like a lab. We're almost like an academic platform, kind of a focused research organization. We work with companies, foundations, academics. You have an idea, you have something you want to test, we're often then there to help you. We can help you develop and actually build your own iPSCs. We can then actually help identify disease signatures in these models, test therapeutics. We actually do help train people, so we're actually open access. So everything we do, we put it online, protocols, we have videos, different languages. We essentially, anything we do, we want to make it openly and available to people as much as we can, and then to really help explore the use of these technologies for iPSCs, testing candidate therapeutics and so forth. So what are we going to talk about today? So we're going to get into glia. So we actually make probably 20 different types of cells in the lab. I'm only going to focus on one today in particular, and that's glial cells. Everyone talks about neurons. Neurons kind of are the things that connect, they fire, but they've got friends. That's the glia. The glia can help the neurons, or they can maybe go awry and they can maybe be toxic or cause damage to the neurons. And that's kind of what we want to focus on, and in particular, oligodendrocytes. Years ago, we got into oligodendrocytes. No one wanted to work on them, so we were like, let's do it. It seems hard to do because they're very difficult to actually make from iPSCs. And so Valerio is really going to talk about this work that he's done over the last few years, kind of how he built the protocols and how we're then going to apply it for this study itself. And this kind of led to an award. Thanks to actually Jeff. He helped push us. We got into this, we submitted it. And if he's watching at home, we hope we miss you. We hope to see you again soon. And I'm going to give it over to Valerio now to talk about kind of CHAMP1, oligodendrocytes, and what we hope to do over the kind of the next months and years to come. Valerio.
Thank you. And that's where I come in play. So I'm basically the friend of the friends. So, so I hope today to kind of give you a different perspective. I didn't come empty-handed. We have some preliminary data, but what I hope to do is to show you what is going to be our work in trying to understand if oligodendrocytes have any role in the pathogenesis of the CHAMP1 related disorder or in the severity of it. So essentially, I'm going to fly through this because you probably have heard it a million times today, what CHAMP1 is and what CHAMP1 does. What I want to stress are the last two parts in which I thought about oligodendrocytes when I saw that there are some cases among the patients of undermyelination or delayed myelination. And this is a process that is underlied by the oligodendrocytes. However, the involvement of the oligodendrocytes in the pathogenesis of CHAMP1 is not yet have been experimentally validated. So why oligodendrocytes? Why they are so important and why they are the friends of the neurons? Essentially, oligodendrocytes are these cells that have the duty of extending this membrane called the myelin sheath that wraps around the axons of the neurons and helps transmitting the electrical signal in the neurons in a steady and fast way. But not only, they also have trophic role, so they secrete little molecules that help the neurons to survive and to work better. Focusing on the origin of the oligodendrocytes, they derive developmentally from some precursors, and some of these precursors, called OPCs, stay also in the adult brain, and they have themselves their own function. In fact, they are also able to form themselves synapses with neurons to help potentiating the neuronal transmission. They also have a role in responding to inflammation, injury, by proliferating and creating new myelinating oligodendrocytes, but also by responding with secretion of cytokines and molecules, everything that helps the neuron surviving. So this is why I hope you understand that the concert of actions between the neuron and the glia, the oligodendrocyte in this case, it has to be seen as a whole in understanding the importance of these cells. So now Tom mentioned that we work with stem cells derived from patients, and when we approach the differentiation of a given cell line from stem cells, we have to keep in mind what is the physiological development of this line, the differentiation. Don't be scared by this. This is just to demonstrate that there are a number of instructions from the progenitors to achieve the finally differentiated cells. In this case, what I want to focus your attention on is the fact that oligodendrocytes derive from progenitors that are in common with another cell type in the brain, astrocytes. And as a result of this, not only we can have overlapping signals, so these molecules that give the instruction to go through each of these phases, but when we approach this simulation in vitro, we can also end up with a very heterogeneous cell population in the end, in which you have your oligodendrocytes, but you also have the co-presence of astrocytes, and of course you might have some of these intermediate cells in the mix. And this was explained in one of our recent publications, in which we exploited one control line that we modified with a fluorescent reporter. And using this line, we have been able to study the in vitro heterogeneity in order to ameliorate existing protocols to create oligodendrocytes from iPSCs and ameliorate it. We came up with essentially this recipe to make an oligodendrocytes from iPSCs. You can appreciate how at each steps, given the molecules that we give or the instructions, we can go from iPSCs to oligodendrocyte precursor cells to myelinating oligodendrocytes. And for each of these stage, we can assess the identity of the cell through different techniques. In this case, what I show you is immunofluorescence, in which we can treat cells with antibodies and see if they express given markers. The ultimate goal is to have the MBP positive cells, MBP being one of the main components of the myelin sheath. Now, once we created this protocol, we always remain with the problem of heterogeneity in vitro. So we found interesting to unveil the different subpopulation through a technique called single-cell RNA sequencing. I'm going fast on this because Dr. Berto, I know, will explain about it. But so essentially, you have a single cell suspension. You can barcode molecularly each cell in order to identify it in a unique way. And once you have these cells barcoded, then you can use some probes that allow us to sequence the entire transcriptome of the cell. The transcriptome being the genes that are actually expressed by the cell. Once we have this immense amount of data, then we can use some models to try and put them all together, and with combination of gene levels, we can identify different subpopulations and study them, quantify them. So it is a way to benchmark the different subpopulations. And this is what we did in another recent publication in which we differentiated oligodendrocytes and glial cells from iPSCs, and we've been able to identify a number of subpopulations, among which, of course, we have our proliferating oligodendrocyte progenitors arriving to differentiated oligodendrocytes, but we also have resting progenitors that we still have to understand what is their role. And of course, we also have the astrocytes, that are the other cell that derive from the same progenitors. So as you can see, starting from the progenitors is also like we can create pathways that resemble the natural progression of what happens in a real human brain. In fact, using a technique called pseudo-time analysis, we can predict what cell derives from which progenitor. And if we set the starting point at the glial-restricted progenitors, we can see that the cells can go in three different ways. In one way, they go towards astrocyte. In one way, they pass through proliferating OPCs and eventually they remain resting as a reservoir, or they can go towards differentiated myelinating oligodendrocytes. Why is that important? Is because we can also quantify and identify the genes that help the cells making this decision. And essentially, to summarize all of this, what we've done so far, we have a benchmark model, and now, given any interference, being that a mutation, an insult, or whatever, we can see how these populations shift, and if they shift, of course. And this is exactly what we mean to do with CHAMP1. So starting from this data set, the first thing that we've done is, where is CHAMP1 expressed? You can see that each of these dots is one cell, and the color intensity is essentially the gene level of CHAMP1 expressed by each one of these single cells. We can also quantify it in a different way, for instance with these dots, and so we can see in each population how many cells express CHAMP1 and how much CHAMP1. So essentially, we found that the three main populations expressing CHAMP1 are the early oligodendrocyte progenitors, the proliferating precursors, and in part also the premyelinating oligodendrocytes, the matures. And now that we have this information, we can also go and look what about the interactors, the known interactors of CHAMP1. And here I show you essentially some of the main interactors. I essentially, from the little I know, divide it into two categories. The first category is the complex formed by CHAMP1, HP1 alpha, POC-Z that are on the left, and then there are some centromere-associated protein that underlie another function of CHAMP1, for instance, in the kinetochore and in the mitosis. What we can see is that pretty much all of these are largely expressed in the three populations, except maybe for the premyelinating oligodendrocyte. So I would say that the most relevant categories that express CHAMP1 and its interactors are the proliferating OPCs and their immediate progenitors, the O2A cells. So this is just a preliminary study that led us to mainly two observations slash hypotheses. The first one is given how CHAMP1 can be involved in oligodendrocyte. So, because OPCs, the oligodendrocyte progenitors, are cells that divide very rapidly, during cell divisions, the cells can accumulate a lot of stress called replicative stress. And this stress essentially induces the apparatus that replicates the DNA to make errors. So the process that we heard before in this series of talks of homologous replication prevents and corrects these errors. But if CHAMP1 is not there, then we are more prone to switch from the homologous recombination to the non-homologous end join that is more prone to make errors. So if these errors accumulate, we might end up either losing some of this OPC pool with a result of less oligodendrocytes, so less myelination, or we can have aberrant oligodendrocytes accumulating that, however, end up not performing myelination properly when they differentiate into oligodendrocytes. Another observation is that because the complex of CHAMP1, POXY, HP1 alpha is involved in the modification of the chromatin. Essentially, just to put it simple, the DNA packs and unpacks in order to allow the access to some factors and some genes to be turned on or off. If this whole complex doesn't work properly, this impedes the progression of the differentiation. As we can see here, along the differentiation towards oligodendrocytes, there are a lot of chromatin modifications, such as trimethylation of the histone 9 or deacetylation. So if this balance between genetics, epigenetics, and microenvironment is altered because we have a less functional CHAMP1 or a non-functional CHAMP1, therefore we can impede at one point, to some extent, the progression of the differentiation towards oligodendrocytes. So just in summary, essentially, these two hypotheses that can be coexisting, in our opinion, can be explained in if there is a failure to perform homologous recombination properly, therefore there is a defective expansion of the progenitor pools, so there is a lack of mature oligodendrocytes, and therefore we can observe hypomyelination. If there is hypomyelination, the neurons are not properly protected and they are more subjected to damage. In mature oligodendrocytes, if we fail to myelinate given parts of chromatin, we cannot have access to stop some cell cycle genes, therefore the oligodendrocyte cannot mature properly, and therefore there might be some myelin instability, some demyelination. So given this background, and I'm going towards the conclusion, what is it our plan to verify if what I said is true? Because it can also be untrue. We are in the process of acquiring patient-derived cells from Coriell, and through St. Jude Children's Hospital, we are going to correct the mutation that this cell line carry in order to create an isogenic control. In parallel, we are also using a control cell line in which we are knocking out the expression of CHAMP1, and then we are differentiating them into OPCs and oligodendrocytes. And then we can perform a number of tests, such as assessment of differentiation through the expression of different markers or gene, morphological analysis to assess the complexity of the cells, or functional tests simply to see if they myelinate. And in summary, the questions we want to answer is, do mutation in CHAMP1 affect the generation of OPCs? Is the pool of OPCs the same with or without CHAMP1 mutations? And if OPCs form, do they lead to oligodendrocytes, and will they form myelin? Finally, another question that we can answer is how these oligodendrocytes interact with neurons that we can likewise form from the same iPSC lines. And with this last set of questions, I would like to thank the CHAMP1 Foundation for hosting us and giving this incredible opportunity. I would like to thank the organization of the conference. This has been really nice, really informative for us. And I'll take your questions.
Who would like to make a question? Okay.
Yes, hi. So have you obtained any IPS lines? And if yes, how many you planning to study?
We just signed the contracts 2 weeks ago, so we're getting the 2 lines from Coriel that they each have mutations. One is the arginine, and I can't remember the other one off the top. I should know. We've filled in. We spent so many weeks and hours doing MTAs, and then from St. Jude's they're going to be the 2 isogenics. So that's what we're getting for now. We're not going to do CRISPR correction, but we have talked with Jeff about there's other lines available that we could potentially go CRISPR correct them ourselves. And we're actually starting to do 1 or 2 of our own lines. We're actually making a CHAMP1 knockout line, and we may also just do CHAMP knock-ins on our own control lines as well that we have that don't have any mutations yet. So for now, yeah, we'll start with those 2 pair and then work our way up.